వాళ్ళు హెవీ అయిపోయింది. ఇంటికాడ టిఫిన్ తిని వచ్చిన ప్రవీణ్ తిన్నాడు మళ్ళీ హాస్టల్లో అందువేళ ఇప్పుడు వచ్చింది హాస్టల్ ఆడ మళ్ళీ ఆడ ఇడ్లీ. Beta lactam inhibitors. So here, drugs acting on cell wall and cell membrane under this category. So now the penicillin is a main example under this one. And here, we have two things here with the beta lactam inhibitors and beta lactamase inhibitors. These are the two. The beta lactam inhibitors for this example under this one is the penicillin. And one more thing is there, beta lactamase. Beta lactamase inhibitors. That means the example is flavonic acid. So flavonic acid is the beta lactamase inhibitors. Beta lactam inhibitors. This is lactamase inhibitors. Right? కొంచెం కన్ఫ్యూజన్ ఉంటుంది బీటా లాక్టమ్ ఇన్హిబిటర్స్ బీటా లాక్టమ్ means simple is the right it is a simple chemical structure and beta lactamase is an enzyme so beta lactam inhibitors generally under this one penicillin and the mechanism action of penicillin is so generally the penicillins are the beta lactam inhibitors it's the drugs okay. So the drugs under this category, basically they will stop the cell wall synthesis or the cell wall replication. Right? So the major role of these drugs is they will destroy the cell walls of the bacteria. And we know that the bacteria cell walls are two types. Already in the last class we discussed about that. The gram-positive bacteria and the gram-negative bacteria. So where the gram-positive bacteria is having the simple cell wall structure and gram-positive negative bacteria having the complex cell wall structure. So this the difference between these two things is due to the having the lipid bilayer where the gram-negative bacteria cell wall. So you can find the right. So lipid bilayer in both sides, inside and outside, right? So two sides the lipid bilayer is there. That's why it provides us some complexity to the cell wall. So it is difficult to destroy that cell wall. Other
Sir, your board is
efficients
not visible.
linked so to destroy that particular right the cell wall, you need to use a very potent or the efficient drugs, right? And where the ground was to back, it's a very simple structure. So means the medium moderate active active drugs also can destroy the cell wall of ground was. So here the mechanism action of penicillin. So if you see this mechanism action of penicillin here, so generally there are three steps are involved in the mechanism action of penicillin. These, right? So the preparation of synthesis or preparation. Synthesis of precursor. Precursor. Preparation of precursor molecules. Right? So precursor molecules. That means, yeah, the precursor. What do you mean by precursor here? Precursor means the starting material, right? Precursor means the starting material. So the synthesis of the starting material. That is the very first step in the mechanism action of this. Okay. So, what are the starting material for this cell wall preparation? What are the starting material? Backbones are the backbones for this cell wall. In the last class we discussed two building blocks, right? The two building blocks for the cell wall vector cell wall cells. Norm and norm. N-acetyl glucosamide, right? We are taking the center of, right? That means N-acetyl glucosamide. Norm is a N-acetyl uramic acid. Okay. N-acetyl uramic acid. Right? So these are the starting methods for the synthesis of that norm and norm. Right? So the first step is the synthesis of precursor molecules, and the second one is the synthesis of preparation. Synthesis of peptidoglycan. Synthesis of peptidoglycan chain. So peptidoglycan chain. But here, where the by combination of these norm and norm, the process one cross-linking one, and they will combine with some other aspects here. Forms you will get a very large gene structure. Combined type one gives a very gene structure, linear gene structure. Other large structure is obtained. It is called the peptidoglycan. Along with this, have some amino acids. Here, okay. So along the so these are the amino acids, right? So the starting materials combines with this particular amino acids here, and it forms a very large structure for the peptidoglycan chain. And this polypeptide is a polypeptide. The polypeptide structure is of the polypeptide nothing but is protein. Okay. Polypeptide one protein cell. And third one is a cross linkase. Cross linkase of peptidoglycan. So cross linkage of peptidoglycan chain here. Okay. So here where this peptidoglycan chain is, so undergoing the cross linkage on the okay. So here the polypeptide, right? The polypeptide chain that is a peptidoglycan chain. It is undergoing some cross linkage and forms a right. So finally by product, it like it forms a cell wall, very rigid structure. Okay? And generally here, this cross linkage is done on the in the presence of other on the surface of the transcriptase enzyme. So this cross linkage of peptidoglycan chain takes place on transcriptase enzyme. Okay? So here, the transcriptase enzyme takes place on the transcriptase enzyme active site. Okay. So this is a key step, right? The cross linkage of peptidoglycan. It's a very key step in the cell wall preparation of the bacterial cell wall. So our drugs, whatever the drugs that take you, like penicillin or the sulbactam or whatever the levulinic acid, right? So they are targeting the transcriptase enzyme, right? It is targeting the transcriptase enzyme, yeah? And it is inhibits. Generally, it is a irreversible inhibition. Right? It inhibits the transcriptase enzyme irreversibly. Enzyme inhibition, there are two types basically: reversible enzyme inhibition and irreversible enzyme inhibition. Right? Well, reversible enzyme inhibition, there are again three types: competitive reversible enzyme inhibition, non-competitive reversible enzyme inhibition, and uncompetitive. Right? Competitive, non-competitive, and uncompetitive. These are the three categories of reversible enzyme inhibition. And irreversible enzyme inhibition, there is no category. Okay. Yeah, the drug chemistry. Right? Majorly, it is the main principle is the right. The drug chemistry is the enzyme inhibition only. Drug receptor inhibition. Yeah. Basically, it is two types. Reversible, irreversible. Reversible enzyme inhibition and irreversible enzyme. So here, reversible enzyme inhibition and irreversible enzyme inhibition. And reversible enzyme inhibition again we consider the three categories: is the competitive, reversible, non-competitive. Uncompetitive. Again, these three categories. Okay. Reversible, irreversible. Reversible again three types: competitive, reversible enzyme inhibition, non-competitive, uncompetitive. मतलब we can get paper 7 also. Enzymes process. Interactive also. Viral drugs also. So, so is mean by computational reversible enzyme? Generally here, the reversible enzyme inhibition means here, inhibition means what is mean by inhibition? Inhibition, stopping. Okay, so stopping. First, inhibition means stopping, preventing. So generally, here the engine, this is the, for example, this is your engine active site. Receptor, we call it receptor here. Every engine having its own cavity or the active site. And right, so there is a inbuilt, right, there is an inbuilt ligand molecule present in the living organism. Then we call that inbuilt ligand is called the natural neurotransmitter. Inbuilt, right, ligand. Whereas the ligand is called natural neurotransmitter. And this natural neurotransmitter is, right, so it is binding to this particular enzymes or the receptor. Okay. So when that natural neurotransmitter is binding or occupying the receptor and active site, then it gets activated, releasing some chemical messenger here, and that chemical messenger is controlling the some biological actions, biological functions, some reactions. That is the usual process which is takes place in the living organisms. Okay. But due to some factors or due to some right, unexpected excitement or the unexpected reactions, right? So where the neurotransmitter working means the functioning will be going to fail. It may be fail its functioning. Sometimes the receptor gets more activated or overstimulated again. It will be overstimulated. In that case, some abnormal reactions will be taken. The abnormal functioning will be taken. Right? The abnormal activities is going to take place here. In that case, where the biological system, or the biochemistry, the normal biological system is going to deviate. Right? The normal biochemistry is going to deviate. So the deviation is called the disease. Generally, the deviation. Okay, the deviation is called the disease. In that case, we'll rectify that system or will correct that deviated system or will bring the deviated system in the normal mode to use the drugs. Okay, that is a external one. This is internal. It is a national neurotransmitter. It is an internal one. Endogenous structure. Generally, we call it endogenous chemical. Endogenous chemical substance. The natural. It is a naturally occurring, which is all present in the living system, right? Endogenous. So, in the abnormal functioning or the failing functioning here, so we, right, so we bring that sort of deviated system in the regular mode with the drugs. Okay. This is the exogenous. This is the endogenous chemical or the substance or the ligands. This is the exogenous ligand, exogenous substance, exogenous chemical. Right? So here, now we have both things, external ligand and internal ligand. Right? Internal ligand already present in the state. External, we are taking that. Right? Now we have only one receptor active site. We have one receptor active site, and the competitors are two. Okay. Like we have one cup, there are two competitors. Okay. Same thing, ఇక్కడ మనకు రిసెప్టార్ ఒకటే ఉంది యాక్టివ్ సైట్, ఇక్కడ రెండు కాంపిటీటర్స్ ఉన్నాయి. ఏది ఎక్స్టర్నల్ మాలిక్యూల్ అండ్ ఇంటర్నల్ మాలిక్యూల్. exogenous and endogenous, or the drugs and gas neurotransmitters. There are two. Both are trying to occupy that active site. Both are రెండు active site లోకి enter అవ్వడానికి ట్రై చేస్తారు. Right? అంటే చేయడానికి మనం కాంపిటేటివ్. That is called the competitive reversible engineering. When both are competing to each other, both are trying to occupy the active site, we call it competitive inhibition. And reversible అంటే ఏంటంటే జనరల్ after entering in the active site, receptor active site, so it will be bind with the active sites. It will be form some interactions, bonding interactions. It forms some bonding interactions, and that bonding interactions is weak bonding interactions. Right? It is a weak bonding interactions. So that weak bonding interactions generally we have three types, like either it is hydrogen bonding interactions, ionic interactions, dipolar interactions, hydrophobic interactions. So ఈ interactions తోటి అది bonding form చేస్తే we call it as a reversible. So it is a temporary interactions, right? Temporary means it is not a strong, weak interactions. Temporary not, it's a weak interactions, right? దాన్ని మనం reversible. అదే irreversible inhibition లో a permanent interactions is forms. A permanent interaction, very strong interactions, and generally it is forms a covalent bond. ఇక్కడ మనకు హైడ్రోజన్ బాండింగ్, ionic bonding, dipolar interactions, hydrophobic interactions. That is reversible. Ivanni weak interactions. Covalent bonding interaction is a strong. That interaction, direct bonding, covalent bonding is strong bond, right? So like, so here cross-peptidase, whatever the pencil, oxy covalent bonding with cross-peptidase. That means irreversibly inhibit the cross-peptidase engine. Okay. Okay. So here this competition. Competition is a competition. Next, non-competitive. So non-competitive again. So here, when okay, so there is a when there is a only one active site and two ligands are trying to occupy, two ligands are competing with each other, then we call it competition. If there is a two, right? If there is a two sorry, what is that? Active sites, right? Two active sites, and the competitors are two. That is called the non-competitive because there is no competition. Okay. Non-competitive means how the means both are occupying the different actors. Generally, in the receptor, this is the enzymes, receptors, right? So all the enzymes are having the additional active sites. Very starting, the very first class we discuss about this. First class we discuss this. And like the enzymes have the active site. The actual active site. This is receptor and the not. Okay. So, enzyme in the additional active site that is called the allosteric site. Additional active site is called the allosteric site. So when the enzyme is having or the receptor is having the additional active site, right? Then here, okay, right? So we have the ligands, right? But ligand That's the limit. Right? We have the drug and reactionary transfer. Here, these two are trying to, when these two are trying to occupy the same active site, then it is called complete inhibition. Okay. And generally here, the drug compound is a external molecule. So obviously it is a highly potent, highly active one. Right? So generally it will be defeat that the internal calcium neurotransmitter, and it is going to occupy the site. Right? ఎందుకంటే మనం ప్రిపేర్ చేసి పంపించింది కాబట్టి జనరల్లీ ఇట్ ఈస్ హ్యావింగ్ మోర్ ఎఫిషియన్సీ ఉంటుంది దానికంటే కాబట్టి అది అదే పై కూర్చుంటారు జరుగుతుంది ఇన్ కేస్ ఇఫ్ ఇట్ ఈస్ నాట్ మోర్ ఎఫిషియెంట్ దెన్ దిస్ వన్ రైట్ the two conditions when the drug is not much potent than the internal calcium neurotransmitter, or if right, if the geometry of the drug is not matching with this then lock and key, according to lock and key model, the geometry, right, the structure of the, right, the shape of the ligand of the drug compound must be matches with the receptor active site. That is lock and key model. Enzyme model is 3 types. That is lock and key model and induced fit model and 3-point contact model. Right? So lock and key model, induced fit model, and 3-point contact. So here the lock and key model explains that when the shape or the geometry of the ligand is matches with the geometry of the receptor active site, then the inhibition is possible. Right? So the primary condition is the geometry must be same. Okay. That is lock and key model. Emily Fisher proposed that model. Then next model is the induced fit model. So induced fit model there is no condition to have the similar geometry of the ligand molecule to receptor active. Right? Geometry similar undas awesome there. But these two models are not explaining the geochemical aspects like enantiomer selectivity and diastereomer selectivity. Right? So the lock and key model and this fit model fails to explain the enantiomer selectivity and diastereomer selectivity of the ligands. That's why the third model is proposed. Third theorem is proposed. That is 3-point contact model. Very, very important. Exam 100%. Right? So 3-point contact model. So as per that 3-point contact model. So here, right, the enantiomers means the enantiomers, right? Generally, the enantiomers we have R and S, right? So both the enantiomers are not having the same interacting points, right? They both are not having the same interacting points. For example, R isomer is having the 3 interaction points. S isomer having 1 interaction with the receptor active site. Okay. So then you can easily differentiate the enantiomers. So the enantiomer selectivity, diastereomer selectivity can be explained by that particular 3-point contact model. Right? Yeah. So here, as per that, right? So here is the non-compound group. When the drug compound is, right? So it is occupying this instead of the actual active site, the drug compound is occupying the allosteric site, right? Instead of occupying the active site, original active site, so it is occupying the allosteric site. Then what happens? Then what happens here? So when this drug compound is occupying allosteric site, so it leads to conformation changes in the geometry of the actual active site. In our the drug compound, so drug compound is occupying the allosteric site. Okay? So generally, the active site, right? Whether national neurotransmitter will be occupied the active site. Okay? It all comes to this allosteric site. But our drug compound is occupying the allosteric site. When the allosteric inhibition is takes place, so it leads to some conformation changes in the active site. Right? So the conformation changes, right? The geometry change out, right? So some geometry అనేది కొంచెం అంటే ఇది ఉందా సపోజ్ ఏంటంటే మనకు ఒక పేపర్ ఓకే అందులో మనం వాటర్ నుంచి పెట్టి ఇట్లా ఒత్తితే ఏముంది దగ్గర అంటే ఏమైంది అది దాని షేప్ చేంజ్ ఓకే రైట్ సో పిండి ఉంటుంది మనం ఇంత ఒక ముద్ద ఉంటుంది దాన్ని ఇట్లా ఒత్తితే మనం అది జస్ట్ కమ్స్ ఇన్ దట్ సమ్ అదర్ స్టేజెస్ లైక్ దట్ హియర్ ఆల్సో when it is occupying this the empty space occupied చేయగానే అది దీంట్లో చేంజెస్ తీసుకొస్తుంది right it is going to close almost it is closed active site ఏమైందంటే దానికి రైట్ అంటే ఏమైంది దాన్ని జియోమెట్రీ చేంజ్ అయింది కన్ఫర్మేషన్ చేంజ్ అయింది కాబట్టి న్యాషన్ న్యూరో ట్రాన్స్మిటర్ డోంట్ ఫిట్ ఇన్ దట్ యాక్టివ్ అది అందులో కూర్చోలేదు the geometry changes, not match, mismatch. The geometry of the national neurotransmitter and your drug sorry, receptor active site. Both are mismatched. కాబట్టి అది అందులో పోలేదు అంటే ఇది మనం డైరెక్ట్ గా కాకుండా డైరెక్ట్ గా మనం దాన్ని అటాక్ చేయకుండా ఇండైరెక్ట్ గా మనం అటాక్ చేసి దాన్ని క్లోజ్ చేస్తాం రైట్ సో దిస్ ఇస్ వాట్ ఇట్ నాన్ కాంపిటీషన్ బికాజ్ దేర్ ఇస్ నో కాంపిటీషన్ కాంపిటీషన్ ఎప్పుడు వస్తుంది రెండు ఒకే యాక్టివ్ సైట్ లో ఉండి ట్రై చేసినప్పుడు when they are both are trying to occupy the same active site then competition arises then we call it competition but here there is no competition but ultimately it is inhibiting the it is preventing to occupy the national neurotransmitter in the active site that is non-competitive. Next uncompetitive uncompetitive ఇక్కడ ఏంటంటే here first it is allowing to occupy the active site right here the national neurotransmitter endogenous chemical first it occupies the active site and forms an interaction with the receptor and there is a receptor ligand receptor we call the engine right engine ligand okay so here is a engine this is your ligand right so engine ligand bonding is formed. The ligand is occupying the active site and it forms an interaction with that. Means your drug compound is allowing to bind with that active site. After binding, yeah, ఇది దాంతో ఆక్యుపై అయిపోయిన దాకా తర్వాత దాని మొత్తాన్ని ఆ కాంప్లెక్స్ ఏదైతే ఫామ్ అవుతుందో ఎంజైమ్ లిగాండ్ కాంప్లెక్స్ ఉంటుంది దాన్ని బైండ్ చేస్తాయి like మనకు topoisomerase enzyme inhibition, topoisomerase enzyme inhibition. There. So like that type of interaction takes place, right? So after binding the ligand with the enzyme active site, then the drug compound is binding with the entire complex, right? The entire complex. So we call that is an enzyme ligand drug compound, ternary complex. The ternary complex. Ternary complex is formed. Means there is no competition. It's allowing to bind with that. After that, it is binding. After that, it is inhibiting the function, right? ఇక్కడ ఏంటంటే బిఫోర్ బైండింగ్ బిఫోర్ ఆక్యుపైయింగ్ ది లిగండ్ ఇట్ ఇస్ బైండింగ్ విత్ దట్, అంటే దాని దగ్గర రానిస్తలేదు, రైట్? అది దగ్గర రానిస్తలేదు, అది అంటే దాన్ని రానియకుండా అది ఆపుతుంది. ఇక్కడ ఏంటంటే ఫస్ట్ రానిస్తుంది, తర్వాత మళ్ళీ ఆపుతుంది అది. సో దట్ ఈస్ అన్కామన్. దీస్ ఆర్ త్రీ డిఫరెంట్ టైప్స్ ఆఫ్. సెవెన్ పేపర్స్ repeated question also. Explain the Indian English. Okay. Yeah. So here, cross linkage of vertical diagram chain done on the constant edge. Now this this right. So the first step and second step. And the third step is a cross linkage of the peptide diagram chain. So this is the how the cross linkage takes place. This is the picture of diagram. Cross peptide is related active site. Active site. Polypeptide. Here, the amino acid residue hydroxy amino acid in the transported nature. So it is binding, it is forming a bond with the D-alanine amino acid. And this D-alanine, further it is forming a linkage, right? So cross linkage with the long chain peptide, polypeptide, lysine and glycine, lysine peptide. So what is happening here? The long chain is combined connecting with this one. It becomes bigger, right? After formation of this one, the polypeptide here, let's say, L-alanine and lysine, lysine, this the alanine combined with the lysine, then lysine, then lysine is and alanine. Also, in this way, this is going to form the polypeptide. Polypeptide form. Peptidoglycan chain. And with the peptidoglycan chain, then these takes place. And it is finally it is modified into cell wall. Okay. So when the penicillin is acting on the right. So here penicillin is instead of this polypeptide chain, right, the penicillin is forming the bond with this. So this is the normal mechanism. The normal mechanism takes place in the but the penicillin involving mechanism we'll see now. So here, of the penicillin structure, the active site CPU is right. So generally here, when that particular peptide glycan chain is occupying the transpeptidase engine active site, so it leads to formation of a right polymerization. And the peptide glycan chain elongation takes place. But when penicillin is occupying the transpeptidase engine active site here, then what happens? Where this active site, right, receptor active site is it is reacting with this particular penicillin. When the penicillin ring, which beta lactam ring opening is taking place, right? And it is going to form this kind of like curve, right? So this the residue is binding with the is reacting with your penicillin. So it is not involving in the peptidoglycan chain crossing because it is already forming a bond with this particular penicillin, right? So this is bond formation of the peptidoglycan chain also crossing is involved in the because already it is binding with this and it is forming a small bond, whether it is irreversible, right? Covalent bond. One is the whatever the new bond forms. It is a forming a covalent bond, and you know that irreversible equation is involving in the covalent bond formation. It is forming a strong permanent covalent bond with the penicillin, right? So already that residue is amino acid active site is bonding with that particular penicillin. So if right, if that polypeptide or the peptidoglycan chain is enter in the that transpeptidase engine active site, right? So no cross linkage is takes place because already it is blocked with the penicillin. I active site blockade. Example blockade is blockade. Okay, so there is no free OH group. Free OH group లేదు కాబట్టి cross linkage జరగదు. Right? There is no cross linkage of the peptidoglycan chain. Then what happens? No cell wall formation of the fatty. Then what happens here? If there is no cell wall, then the pathogen cannot be exist, cannot be survive.